spermatocytes. Because these cells will be very different than the rest of the cells in the body. So the immune system could potentially attack them. All right, so this is a barrier for that. And it's kind of interesting how this really works. I was always trying to figure out, do these just allow passage? Literally what they do is these spermatocytes under spermatogenesis migrate from the perimeter into the lumen here. And basically, if this is a bridge like this, and here's those primary spermatocytes, no flagellum yet, they will form another bridge like this, and here, which then seals that off, and then the other bridge just basically disintegrates. And now they're in the lumen here. So that's how you can actually get passage and always maintain that barrier. Okay? So then this just shows you here the process of this. We have spermatogonia, diploid cells. They undergo mitosis to make more spermatogonia. And that's important because in the males, when we made those 100,000 sperm in that 10 seconds, that came from how many spermatogonia if we made 100,000? 25,000, because you get 4 per spermatogonia that goes through this process. So you have to regenerate spermatogonia. So what happens is they go through mitosis to produce more themselves. Not usually talked about, but that has to happen. But some of them undergo the S phase, and they're going to undergo meiosis. Okay? And once they've done the S phase, they're now 2N4C. We call those primary spermatocytes. 2N4C, just like we've talked about repeatedly with meiosis. We go through the first meiotic division. We form 2 cells. Each are 1N2C. Those are called secondary spermatocytes. They're haploid, but they do have 2 pieces of DNA double strand for each chromosome. So we have to separate those chromatids in the second division. That's when we end up with spermatids. They're not called spermatozoa yet because they're immature. Things have to happen to them, like adding the flagellum, adding the acrosome. So there's a maturation process that will occur. And in humans, that's going to occur in the epididymis, okay? And what happens is, as I mentioned, they migrate to this lumen, and this lumen gets filled with fluid. So it's just like if I had a tube and I had lots of fluid at this end, they will be propelled by that fluid to the other end of the tube. And where that other end leads to is the epididymis, okay? So all these tubules eventually lead to the epididymis. That's how the sperm get there. They don't swim there. They're just through bulk movement. All right, so let's look at some images here. This, again, just shows you the 2N4C, 1N2C, 1N1C. Here shows you mature sperm. So it has the acrosome with specialized digestive enzymes, etc. There's mitochondria here. There's this flagellum. And this shows you cross-section through a, so this would be the actual testes. So here's a tubule. And I don't know if you can see this in connective tissue here, but here's interstitial cells between a tubule. Another one there. And that's where we have the Leydig cells or interstitial. Okay? And you've had this repeatedly in the 1A lecture. And that is you've had repeatedly that you have the interstitial cells or Leydig. I don't know if you've been given the names. I can't remember. What we have here is conversion of cholesterol to testosterone through the Leydig or Sertoli cells. Sorry, Leydig or interstitial. And then that testosterone gets converted to DHT by the Sertoli cells. And those Sertoli cells, remember, are the same cells that are providing this bridge. Sometimes they're called nurse cells. So within this, here's the interstitial cells producing the testosterone. We then have conversion by the Sertoli cells here. And I would not ask you on a lab exam, I would not have a cross-section here and say identify the Sertoli cells. It's just not possible, okay, at the time span, okay? But you might see some cells that have condensed chromosomes. So I would potentially ask you, is this undergoing meiosis? And you'd say yes, because you see condensed chromosomes, okay? Here it just shows you interstitial cells here. Here it shows you a close-up. You see the flagella here. So in this case, for the rat, within the lumen here, they do have flagella attached, but they're not fully mature. It occurs in the epididymis, okay? Any questions about spermatogenesis? Okay? And remember, it's fairly equal cytokinesis, and from one spermatogonia that undergoes this meiotic process, we yield 4 spermatids and then 4 sperm. In terms of oogenesis, we're going to use similar terms, okay? We'll do the rat first. The rat's very typical, what you're used to, but with humans, there are some differences that I'll point out. Okay, in terms of the ovary here, we have ovary here, and I mentioned that we talked about this growing follicle, okay? That growing follicle is referring to the oogonia and the resulting oocyte that's being produced, and the increased number of cells surrounding it, okay? So those increased number of cells are going to be granulosa cells. So when we start off with this oogonium here, pre-birth in humans, that would have undergone mitosis to form, maybe 1 million oogonia, but at birth, 400,000 left at puberty. So when she's born, all of her oogonia have undergone this, and they're all stuck in prophase 1 of meiosis. So they're all stuck as a primary oocyte. So it's the same process: spermatogonia, primary spermatocyte, second spermatocyte, spermatids, sperm. Here we'll use the term primary oocyte, secondary oocyte, and ovum. So in a rat, it's very typical. They have the primary oocyte, then they go that completion of the first meiotic division, yield 2 secondary oocytes, but it's unequal, so they call this a polar body, and this is secondary oocyte. Vast majority of the cytoplasm ends up in this cell, not in this, okay? And this is not drawn to scale by any means. The difference there is more like 40,000 times, okay? It's a pretty dramatic difference. And then the secondary oocyte undergoes the second division to yield another polar body and the ootid, technically ootid, and maturation to the ovum, okay? Unequal. So a rat, when she ovulates, she does release true ova, multiples, because she can get implantation in each uterine horn, okay? Any questions about what's happening here? So that's in a rat, okay? Pretty straightforward. Yes, question? So the question is, could rats potentially have semi-identical twins? Is that what you're asking? Conceivably, yes, okay? Yeah, conceivably. Now, I don't want the lecture to focus on semi-identical twins. That wasn't the purpose of that. The purpose was to show you how to integrate a conceptual question. But yes, no more. So in terms of rats, all the meiotic divisions are done before this fertilization. In terms of humans, it's different. That's what we're going to do next, okay? So the assumption is that since we're placental, we can indeed have a fusion here of the second polar body, and since we'll eventually get implantation, it might be viable. But something like a chicken embryo would not happen that way because you wouldn't have enough nutrients for that, okay? So you can look at the sort of development of organisms to be able to answer that question yourself in terms of generally, okay? Not precisely. So in terms of humans, this is the difference of humans. They're all stuck primary oocyte at birth. Ovulation, when the woman reaches her puberty and she goes through a menstrual cycle, a few of these primary oocytes start to develop in response to the hormonal changes. But only a few, okay? And eventually, typically one wins out. What happens is she then produces a second oocyte in the first polar body. It is a secondary oocyte along with the surrounding cells, in that case granulosa cells, the zona pellucida, that gets ovulated. It is not just secondary oocyte. It is going to be that secondary oocyte, that zona pellucida, and all those granulosa cells, okay? And I'll show you an image of this shortly. So then what happens is we then have a sperm come here, the membranes fuse, And that's where we start the first events of fertilization. So in response to membrane fusion, there's lots of changes in metabolism, waves of ions, racing like that. And eventually, the sperm nucleus literally gets pulled into that one secondary oocyte. That's going to trigger the second division of that nucleus. All right? And then we'll effectively have 3 nuclei in here. One of them gets pushed out through cytokinesis to form a very small second polar body. And then we have these 2 physically separate nuclei. Okay? And that typically we call a heterokaryon. The nuclear membranes break down, but now that DNA is in the same kind of cytosol, so it's now true diploid. Okay? So that's what happens in humans, all right? Does that answer the question? All right. So let's go over this image here. This image will clarify what gets released during ovulation. So here's this oogonia starting the maturation process. Maturation, going through this process, we increase the number of cells surrounding it. Okay? So we end up with this growing follicle. So if you look here, it has the actual, in the case of a human, a secondary oocyte here, in the case of a human. Let's just do the rat for now because it's easier. So we have the ovum here, and then there's a extracellular matrix out here called the zona pellucida. And then we have granulosa cells here, and as part of the proliferation phase, we increase the number of the granulosa cells. That's why the amount of estrogen is increasing during the menstrual cycle in humans, because the number of cells are increasing. So each cell itself is producing less estrogen due to the inhibition, the feedback. But since you have more cells, the levels of estrogen overall can start to increase. Okay, so that's what's happening there. So we have those granulosa cells. We have some out here. Plus we have cells further out here that are modified called thecal cells. The thecal cells are analogous to this. What the thecal cells do is convert cholesterol to testosterone. What the granulosa cells do is convert the testosterone to estrogen. So those would be analogous structures or cells. Okay, so I could certainly ask you a question like that on the exam. All right, I could say, what's analogous to Sertoli cells in a female? And that would be the granulosa cells there, okay? So what gets released in a human would be a secondary oocyte, zona pellucida, and these surrounding follicular cells. So the sperm have to get through these surrounding cells so they can touch that membrane. It actually takes many sperm to do this. So many sperm are actually approaching that egg. Many sperm are digesting some of those follicular cells. They tend to sort of target one spot. And what happens is, after multiple of those have now reached this particular location, you start to degrade the follicular cells, and then you have a pathway for the next sperm. So the first sperm aren't typically successful in fertilization. It's usually subsequent sperm. And Dr. Firestone has gone over this a lot. I don't have time to go over that. Make sure you're familiar with things. Again, in humans, it's that secondary oocyte that gets released. And then the remaining cells, in this case, these granulosa cells and some of the thecal cells that remain behind, they form a structure called the corpus luteum. And they continue to increase in numbers, and they make that estrogen and progesterone. So that's what's maintaining the estrogen and progesterone here, is that corpus luteum. But it has a half-life, okay? Meaning after a few days, it will degenerate and no longer be able to make those hormones. And that's what resets the cycle, okay? Now, this argument about when does life begin, is it at the moment of fertilization, or that, to me, is such a trivial question, because are the sperm alive? Yes. Are the eggs, secondary oocyte alive? Yes. So life was way before then. I think the real argument here is, when do you get a zygote, because that can undergo mitosis. The gametes can't undergo mitosis. It's the zygote that does that. So the real question isn't when does life begin, it's when you get the special ability to undergo mitosis, right? And that's what the whole debate really is about, okay? All right, so here shows you a ovary. Realize this has many different follicles in it at different stages. There are thousands of oogonia in here, and only a few have responded. Now, in the case of the rat, she can easily ovulate, 12 ova per ovary, paired ovaries to the fallopian tube and the horn. So what we see here is multiple ones of these at different stages. We don't see the developing ovum or oocyte here because it's out of the plane. So it's just like this. If we have something large like this and I do a thin section through it, I may not catch the nucleus. I may not catch the oocyte developing. Okay? It's still there, we just don't see it. So this is the fluid-filled space, the antrum. Here's granulosa cells here, and the outer cells here that are darker like this. You can see the difference. Those are the thecal cells. This was the drawing trying to show you thecal cells here, granulosa, and they do actually look much more different in the actual specimen. So granulosa cells here. Here's the zona pellucida, the clear white spot there. That's very obvious. We haven't formed enough of an antrum here to see that space yet, and then we don't see that developing oocyte in that cross-section. Okay? Again, just shows you that. Here's more of a typical looking one. So here's the antrum fluid filled. Because it's being filled with fluid, these actually migrate towards the surface of the ovary. They get enough fluid filled that they rupture. And when they rupture, they release this, in the case of a rat, the ovum, in the case of human, the secondary oocyte, with the zona pellucida and the surrounding granulosa cells. So those sperm would have to swim through there for get past those, and they eventually hit this egg membrane. So they have to go through the zona pellucida as well. Okay? And just be familiar with corpus luteum. And this shows you an illustration of this. You can see how large this is. Many cells to continue to produce that estrogen and progesterone to maintain the endometrial lining, to maintain the cycle. We're not going to have time to talk about fruit flies, Drosophila, that. There's a ton of work being done in development here in Berkeley. And I just want to point out one thing. So if you look here, this stain is specific for a type of messenger RNA called Bitcoin. And the only purpose of this slide is one, and that is during maturation, we set up things like gradients. So we set up gradients of mRNA, we set up gradients of proteins, we set up gradients of hormones, we set up various gradients. And those gradients can be used to determine anterior versus posterior, triggering transcription factors, etc. Whole thing here, as during maturation, we develop gradients. Okay? So during oogenesis, oocyte, there are gradients within that egg. Okay? So I talked about fertilization, a couple events. So there's clearly the union of membranes and then the union of nuclei or DNA. There's external fertilization. That is away from the body of the organism, okay, effectively. Okay? So it requires a moist environment, large number of gametes, usually low fertilization rate, and certainly increased morbidity of offspring if there's no protective mechanism. Question? So the question was, do the thecal cells get released with that oocyte? Typically, it's just the granulosa cells. The thecal cells remain behind. It's those granulosa cells that immediately surround that developing oocyte, okay? So internal fertilization, you create a moist environment by having internal fertilization. It's typically, you don't need as many gametes, but you need to have copulatory behavior and some delivery mechanism and then storage of gametes, okay? So humans have internal fertilization, although really it's not because the sperm have never crossed any membrane except for the egg membrane. But it's considered internal because it's occurring within a cavity of that female. The cavity being vagina, the uterus, etc. Okay? So it is considered internal because it's within the body. Okay? There are some organs that literally have internal fertilization where the male will take their copulatory organ, penis typically, and they literally inject their sperm through the body of that organism. And some of the first attempts at in vitro fertilization, and that with humans, was they literally take and inject sperm in the abdominal cavity trying to hit the fallopian tube. Very early experiments in this stuff. They have much better methods now, okay? Advances in science are great. This is a time course. Okay, a lot of people fail to understand this is a time course, so we're going to go from left to right in terms of time course. So this shows you for sea urchin. The outside yellow here is the jelly coat that I mentioned. Here's the egg itself, has the nucleus within here, the true 1N1C. Preformed as part of that maturation process are these cortical granules. So they're vesicles outside at the perimeter. They're preformed with various proteins, enzymes, etc. And what's going to happen is the sperm will come along here with their acrosome. They digest this jelly coat. And then we have this other membrane here called the vitellin layer or vitellin membrane. And there's receptors from the egg here. Sperm come through. This is important because externally, there are thousands of different species of sperm that potentially do this fertilization event. So externally, in this case, you need to have some sperm-egg receptor recognition because of all the different species. So we have the touching of the membranes. So let's talk about what happens. Normally, the membrane potential here is negative. Okay? So what that means is the normal charge here is negative relative to the outside. And there's been a lot of energy spent to maintain that. Okay? So there's a little negative charge here, maybe about minus 40 millivolts. But remember, this distance of this membrane is 10 nanometers. So that's a voltage difference of, sure, only 40 millivolts, but it's only over 10 nanometers. If it was a AA battery, like you're kind of used to, it would be about 25 volts. So pretty decent voltage there, actually. So what happens is when we have this first interaction like this, one event is sodium comes rushing in. Sodium concentration is much higher outside than inside this egg cell. As a result, once you open sodium channels, sodium rushes in, and this membrane potential changes dramatically. It becomes a plus voltage. It went from minus to plus within milliseconds. Within milliseconds. That's a huge change. It'd be like you driving 100 miles per hour, slamming the car in reverse, and doing 100 miles per hour in reverse in 3 milliseconds. Huge change. As a result, all these proteins completely change their conformation. And as a result, all these sperm receptors look drastically different. So no other sperm can bind. All those proteins take these weird conformations to this new environment, but that would be lethal if it was kept very long. So this reaction, the fast block, occurs within milliseconds. And that's important because there are many sperm out there, and we want to have only one successful sperm. Okay? So the fast block happens within milliseconds. That triggers a reaction. The wave of that change literally goes from this point and goes around the perimeter. So if this was that, and this is the point of sperm penetration, we'd see a wave go around this, literally. And if you do studies with dyes that indicate the voltage, you'll see the change in color. Okay? But that can only occur for a short period of time because it would be lethal. So then we have what's called a slow block. In response to the changes in ion concentration, this membrane potential, these cortical granules fuse with the membrane. And when they do that, notice they dump their contents into the space between this vitellin membrane here and the egg membrane. So this membrane is separating. It's expanding away, and it gets very hard. So there's these proteins in here, et cetera, that change the structure of this. It separates the membrane, and now this entire thing is called the fertilization membrane. And that prevents additional sperm from coming on board. So there's a fast block and slow block. Humans don't appear to have the fast block, but humans certainly have a slow block. Okay? Any questions about this? And this is the time course, from left to right. So in the lab, what you'll see is you'll take and have the eggs. You'll have the sperm. You'll see what they look like. You'll add the sperm. The sperm are small enough, but if you set cold elimination, you can see their flagella in that. Then you'll see this wave. And you'll see this start to form at one portion of that egg, and you'll see that membrane separate and move over as a wave over the entire thing. Okay? Just watch. It takes about 2 minutes at most. Okay? So that's the slow block. Fast block resets itself. You get the normal membrane potential. The metabolism of the cell is fine then. Question? So the question is, would you have twins if 2 sperm came in? No, you'd end up with a triploid organism, right? Because you'd end up with 3 sets of genetic material, and that would typically be lethal. Okay. Also, since these things, so we don't typically get, so we don't typically get those sorts of events to be viable. Okay. All right. There are some examples of partial triploids. Certainly we've seen with Down syndrome, things like that. Okay. So any questions about this? Yeah. Feel free to ask questions. Now this shows you what it looks like for a rat or for a human. So here's that egg. Here's the preformed cortical granules. Here's that zona pellucida. Here's all these follicular cells. Here's the sperm having to digest away some of these cells. And then here shows your reaction here. And then we don't get the fast block here because internal fertilization is typically the number of sperm that would be doing this at the same time is very low because we don't have the same number of sperm as external fertilization. And then what happens here is the sperm nucleus gets taken up. And then here's the rest of that sperm. The rest doesn't come in typically. Mitochondria don't get taken up typically. Here's the cortical granules, so we start to do that slow block. So it takes several sperm to get through those cells, but typically only one that's effective at fusing the membranes and then the genetic material. Any questions about that? Okay, so let's just show this represents the serous eggs unfertilized. Here they're fertilized. These look drastically different. Okay. If you had done a time course, you would have seen this membrane separate. Let's say the sperm entered here. You'd see the separation here. It would eventually spread around to the entire structure had that fertilization membrane. So this just shows you those cleavage reactions. So starting on the left, that's a sea urchin egg that's fertilized. I definitely probably will have some illustrations of this and ask you has fertilization occurred or not. Various questions like that potentially. So fertilization has occurred. Clearly the slow and fast block have occurred. Here's a 4-cell stage. Here's a multi-cell stage. Notice there's a cavity forming internally. This would be called the blastocyll. Cyll for cavity, internal cavity, blasto for the blastocyst stage. And that allows a space for cells to migrate inward. And I forgot balloons, but usually I bring balloons and blow them up. You can imagine taking your finger and pushing it on the balloon. So that would allow cells to migrate from the outside in. They're literally motile now. They migrate from the outside inward and forming their cantharon, the primitive gut, and start to form the third germ layer, the mesoderm, and the endoderm. Okay. 2 terms that refers to how you cleave this. Holoblastic cleavage refers to the first few divisions where you cleave the entire egg, or the zygote in that case. Okay. So if you notice the 4-cell stage here, 2-cell forming, 4-cell, they go through the entire amount of yolk. So whether an organism is holoblastic or meroblastic, where they just do part of it, it's usually dependent upon the amount of yolk they have. Okay. So when we talk about a chick egg, which is a huge amount of yolk, right? We're going to have the divisions occur at a very small portion of cytoplasm up here. So that would be a great example of a meroblastic cleavage. You can't cleave through all that yolk. There's just too much.